Okay, in this video, I'm going to talk about the importance of optical isomerism with biological molecules. So whether you do biology or not, you do need to know this. Okay, so here I'm not doing here. This is a test tube. Okay, and it's nothing to do with living things. Okay, and when you do it, what I've got in there is I've got butan 1-ol, butan 2-ol, sorry, and butan 2-ol has got an asymmetric carbon atom, which is there, 4 different groups attached to that carbon, an OH, an ethyl, a hydrogen, and a methyl. Okay, so we oxidize that by adding potassium acidified potassium dichromate. That's that orange stuff there, K2, sorry, not KR, K2Cr2O7 plus some acid in there, okay, and you will oxidize it to butanone. And of course, the dichromate will go green because it's been reduced to the Cr3 plus ion. Right, now, as far as this reaction occurs, it doesn't matter which optical isomer of the butan 2-ol you have got. Both of them react equally well, and you'll end up with exactly butanone. You'll end up with exactly the same product. Okay, so when you're doing a reaction in a test tube, usually it doesn't matter which optical isomer you've got; they're both reacting exactly the same way. Right, now it's different if the reaction is catalyzed by an enzyme. Okay, so here we're going to look at, I'm going to rub that out in a minute. Okay, here we're going to look at the oxidation of lactic acid. Okay, and that is lactic acid. Okay, and that is oxidized by an enzyme to pyruvic acid, which has got this formula. It's a ketone. Okay, so you've got a secondary alcohol going to a ketone, but it's catalyzed by an enzyme. And actually, the oxidizing agent, if you do biology, the oxidizing agent is NAD+, which you'll probably come across later in biology. Nicotinamide adenine dinucleotide, that's the oxidized form of it. Right, so now this does matter which isomer of lactic acid you've got. Only one of the optical isomers will react with the enzyme. Okay. Now we need to see why that is the case. Okay, so here's the enzyme. I've drawn that in light blue. Okay, and there's the surface of the enzyme. One minute. The surface of the enzyme, and I've got two of the both isomers. I've got this one, okay, and I've got its non-superimposable mirror image on the right. Okay, so you see I've reversed the positions of the purple hydroxide group and the purple hydroxide group and the blue methyl group. Now you can see with this optical isomer, it can fit onto the surface of the enzyme. So the blue group falls into the special hole, which is the right shape for that. The purple into the purple hole, the green into the green, and so on. However, you can see with this one, no matter how you play around with that, you would never get the colors to match up. You never get the group to fit into the right hole. So you can see that this isomer will not react. So as far as the enzyme is concerned, this isomer just doesn't exist. It can't see it. It's completely blind to it. So that is why, generally speaking, in biological systems, you will only get one. If you've got a molecule which has got an asymmetric carbon in it, you will only get one of them. You will not get the other one. Okay, so yeah, this only one of the isomers can bind to the enzyme. The enzyme is essentially blind to the other one. Right, let's just have a look at amino acids, okay, which you've come across at GCSE in biology, right? Amino acids have the following general formula, okay? They have, you have a carbon atom with a carboxylic acid group attached to it. You have a hydrogen attached to this carbon atom. You have an amine group attached to it. And then you have this group, which you're going to call R. Now, there are 20 different types, okay, of naturally occurring amino acids, which are used to make protein. And so you've got 20 different versions of what R can be. Right, so I do the, so the two simplest amino acids. Okay, so the simplest of all is when the R group is a hydrogen. That was called glycine. And the next thing that R can be is a methyl group, CH3. Okay, now you can see that in this case, this carbon here is asymmetric. Okay, so in alanine, it has an asymmetric carbon, so there should be two versions of alanine, but you only ever find only one isomer exists in nature. Now, glycine, the reason why I put glycine here is because this carbon in glycine, you can see, is not asymmetric because you've got two hydrogens attached. But that is the only naturally occurring amino acid which is not, which doesn't have an asymmetric carbon in it. So that doesn't exist too, but that's the exception. All the other 19, 18 amino acids all exist as can exist as two isomers, but they only exist as one. Okay, so in nature, there's only one version of alanine. That's the alanine, the version you've got in your body, in all the proteins in your body. Same proteins in a tortoise, in an oak tree, in a bacteria, in a fungus. Everything in nature uses the same version of alanine, only one version. If you gave somebody some synthetically made alanine of the other optical isomer, then your body just wouldn't be able to use it and you would excrete it. It wouldn't be able to be used because it wouldn't, the enzymes, it wouldn't fit into the active site of the enzymes. It just wouldn't work. Okay, so in biological systems then, it's usually only one of the optical isomers which reacts, which is not the case in non-biological systems. Very often, usually you end up with a mixture of both isomers and they react in exactly the same way. There's another little way to illustrate this, and it's a molecule called limonene, which has the limonene. It's found in citrus fruits, and it's got the following formulae. Okay, so you have a six-membered carbon ring with a double bond in it there. So that would be cyclohexene. It's got a methyl group there, and then it's got this alkyl group attached to that carbon there. Okay. Now you can see that, right, so these carbons, CH2, so they're not, you don't have four different groups, that one as well. These, of course, have only got three groups attached anyway, so they can't be asymmetric. That one's the methyl, so it's not asymmetric, but this one here, the purple one, is asymmetric. Okay, so that means there are two versions of this molecule called limonene. There's two isomers. Isomer 1 and its mirror image, isomer 2. Now, the one that occurs in nature, isomer 1, actually that tastes, you can taste it, and that tastes of lemons. And that's because that molecule, it fits into the it fits onto the surface of your taste receptors, and it's the right shape for it. It can bind, and it will trigger a nerve impulse, and that's the taste of lemons. This isomer will not bind to the same receptor, okay, because it'd be the wrong shape. And this one actually tastes of menthol, okay, tastes of menthol because it doesn't bind to the same receptor. Okay, so this whole idea of asymmetric molecules binding to the surface of an enzyme or not, it's a bit like if you put, I've said your hands are mirror images, non-superimposable mirror images of each other. If you get a glove, the left-handed glove will not fit the right-handed glove properly. Your thumb will end up in the wrong place, won't it? You know. Okay, so let's leave that one there.